Our next speaker is Dr. Gary Chastagner from Washington State University. Gary visited on a number of our farms this past summer in all of the regions of the state, actually. Fairbanks, Matsu, Kenai, and all the way down to Homer, I believe. Yeah. And he's a plant pathologist. And he was checking out all the little nasty diseases that may be growing on our peonies, and took samples and did a bunch of studies, and he's going to present some of his findings for us this afternoon. So if you would welcome Dr. Gary.
Thanks, Harry. Can everyone hear me? Well, it's a pleasure to be here. Nice to see bright sun. A lot of times in Western Washington, we're overcast. So as Harry indicated, what I would like to do is talk about some of the survey results that we have, and particularly as it relates to botrytis, but I'll also talk about a couple of other things that we observed. Before we get into that, I'll just give you sort of a brief overview of sort of my research program at Puyallup. That way you'll have an understanding of what I'm about and some of the things that we do, and then I'll go into the other parts of the presentation. Washington State University, the main campus for the university, is over here in Pullman. I'm located at a research and extension facility that the university has in Puyallup. This used to be a fair, there used to be a fair amount of agriculture in this area. It's mostly we're growing warehouses right now and houses and stuff. Mount Vernon, where there's quite a bit of commercial flower bulb production and cut flower production, would be up here closer to Canada. And my program is actually quite varied. I was hired back in 1978, actually, to work on ornamental bulb crops and turf grass. So I still do a lot of work on ornamental bulb crops. I don't do any work on turf grass, but now I also do a lot of work on Christmas tree diseases, conifer nursery stock, and nursery stock where sudden oak death, which is an invasive pathogen. So we have quite a large program as it relates to that. This is a picture that I took in the Puyallup Valley, not too far from our research center. Historically, this was a major ornamental bulb production area, particularly daffodils, Mount Rainier in the background, and stuff. This land has all been sold, and warehouses will be going up on it. We're very fortunate at our research center to have quite a bit of land available to us to do research trials. So each year I do a number of field trials relating to ornamental bulb crops. We also have some nice facilities as far as hot water dipping tanks, where we can do trials relating to the use of hot water dipping tanks to control diseases on bulb crops. And I'm very fortunate to have a very good staff that works with me, and a number of graduate students as well. Specifically in my ornamental flower bulb projects, we're looking at some molecular-based assays to detect and quantify soil-borne pathogens, particularly on tulip and iris. And then we're working here a few years ago. Pat had contacted me about being involved with some peony work on botrytis, and that grant didn't get funded, but we've still continued to move along with that. And so we're doing some work to look at the identification of botrytis species that are occurring on peonies and maybe how to manage them. And then have a number of other things. We're looking at the prevalence of virus diseases on lilies and dahlias at cut flower farms in Western Washington. There's a lot of small cut flower farms to supply cut flowers to farmers' markets. I'm very fortunate in that I have a number of different grant sources. Most people don't understand how universities work. They think that since you're a university professor, the state's giving you lots of money to do things. I get about $1,500 a year from the university to run my entire program. And I have to pay for my phone, the paper I use, everything now. So it's changed a lot since I was first hired. So really, the programs that people like myself, Pat, have are really dependent on grant money at this point in time. And so fortunately, the Alaska Peony Growers Association were able to get some funding, I think through a specialty block grant program, which that program, if you ever get a chance to talk to your congressman, make sure that they understand how important that program is. It's important up here. It's very important in Washington. I'll talk about the survey. So there was this article, probably most all of you have seen it, in the Alaska Airlines magazine in August about peonies from Alaska, the last frontier, and about this new export crop. And I can tell you this, that one of the things I was impressed about in July when I came up here for doing the survey is the enthusiasm that there is for growing peonies. It's tremendous. This is a quote out of a preference that Alan Armitage wrote for a book on the diseases on herbaceous ornamentals. And unfortunately, when we have vegetatively propagated plant material, we tend to have the potential to have a number of diseases that can move with that material. And in some cases, some of these cut flower bulb crops tend to have some fairly significant diseases that will build up over time in plantations if they're not properly managed. So the key thing here that, as Alan says, is it's not a question of whether you're going to have disease. It's, do I know how to identify, prevent, or reduce the damage? That's the key. You may start out with a few plants, not have any problems, but as you begin to scale up, you're going to see more and more problems. There are a number of diseases potentially on peonies. This list could be much larger than this, but the reality is there are some that are fairly common that you have to deal with, and one of those, of course, is Botrytis. There are some peonies that have powdery mildew issues. If you have poor soil drainage, you can have Phytophthora root rot issues, and then there are some viruses problems. And as Richard said, there's some nematode issues. If you have warm soil conditions, you're not likely to maybe have some of the nematodes, maybe any of the nematodes. And then there are these, what pathologists like to refer to in horticulture as physiological problems. We don't really understand what's causing them in many cases. So when we can't identify a pathogen or an organism associated with, we say it's an environmental or physiological type of a problem. That just means we're copying out, we don't know what we're talking about. So in the case of Botrytis, there's about 20 species of Botrytis as a pathogen. It's a fungus. So if you go to the grocery store and you buy your mushrooms, you're buying a fungus there that's an edible one. These are microscopic fungi. And if you look at this, this is a genetic map of the relationship of different Botrytis species. And so, for example, Botrytis cinerea is here, and its host range, it says polyphagous. That just means it can attack a large number of different plants. It's very common on grapes. It's very common on strawberries. It's very common on many ornamental plants. And it can occur during production, and it can cause post-harvest losses as well. Part of the reason that it's important to manage diseases for inoculum levels in cut flower fields is to reduce the potential for post-harvest losses when you're storing and shipping flowers. If you look here, though, at most all of the other hosts and the species for the other hosts, they tend to be very narrow and tend to only occur on certain hosts. And it turns out that Botrytis cinerea, although it's very common, and here's a Botrytis pseudocinerea, the only difference here is this one is a natural population that has natural resistance to certain fungicides. But both of these have a very broad host range. These others have a very narrow, defined host range. And most all of these other species are very aggressive Botrytis species. They can attack uninjured tissues without nutrients and things like this, whereas Botrytis cinerea will commonly come in when you have conditions that you have severe stress on the plant, or you have injuries, hailstorms and things like this, or after the flowers bloom, the pollen from the flowers can get on the leaves and provide a nutrient source for the botrytis to grow on, and that favors the development of botrytis cinerea. For example, on ornamental bulb crops, there's a number of these botrytis diseases. This is tulips, can get on the foliage as well as the bulbs, lily flowers and plants, and daffodils. And these are very aggressive diseases that in the cases of the bulb industry are commonly referred to as fire, because you can go out there in a field and when you first see it developing, a couple weeks later the entire field can be killed. It's like someone went through there with a blowtorch. So these are major problems for ornamental cut flower producers. So what about the situation with peonies? So when you look in the literature, it says that Botrytis cinerea, this name Botrytis is the perfect, the name of the perfect stage of this pathogen. So Botrytis cinerea and Botrytis peony. Well, right away, Botrytis peony tells you something. This is a botrytis that was found on peonies, and it attacks peonies. And so it tends to be, it's thought to be host-specific. So these are the two botrytis species that you see in the literature. This grant that Pat put in a few years ago was not funded. She and I were talking, and I think 2 years ago in 2012, I said, well, if you can get some samples sent up to me, we'll do some isolations and see what we can find, see whether we have cinerea or peony or both, or maybe something else up here. So a few growers sent us some samples, and we found some things other than cinerea and peony. And so then the peony growers up here got some additional funding, and so we did this sampling this last year. As Harry indicated, we visited farms up in the interior, in the Matsuk Valley area, and then down in Soldotna and Homer. And I did this visit between July 15th and the 22nd. So what was unusual about last June and July? Hot and dry. Okay, so would you expect much botrytis? No, you wouldn't expect much botrytis, because most of these fungal pathogens that attack the foliage or attack the flower, they require periods of free moisture for the spores to germinate and infect the tissue. So if we have periods where we don't have that free moisture, we tend to have less disease pressure right away. These just show, I visited 25 farms. We also visited some farms in Western Washington as part of this project to gain some additional information about what's going on in Western Washington. So I was looking up some differences in environmental conditions between Western Washington and Alaska. This was just one that really stuck out, frost-free period in days. So Seattle, about 200 days with no frost. If you're down here in Fairbanks, you're about 60 days with no frost. Big difference, obviously. I was a little surprised by the precipitation. Seattle gets around 30 inches of precipitation a year. We have virtually no precipitation during the summertime, so we're quite dry during the summertime. And of course, when October, November rolls around, then we have constant drizzly rain events and things until June. Whereas when I was looking at Homer, it was fairly dry in July, or June and early July, and then it picked up quite rapidly there in late July and August and into September. So you can imagine that under those normal conditions, that would be extremely favorable for the development of botrytis later in the season. So these are just some pictures of some of the farms. They range from relatively small farms to fairly large. Some people without just soil, and others with a weed barrier type. Number of people mulches down there, and then there were a couple of Weeder geese farms, which will be on the agenda. I was really impressed by that. Just another picture of one of the farms. Some really nice plants. I will say that I've been on some farms in Washington, and my general impression is we had a lot more variability in the plants and actually quite a bit more disease than I saw in a number of the farms up here. What did I actually see? One of the things that stood out is there's a number of plants that had what I would consider to be virus or virus-like symptoms. This is a picture of a virus infected plant. I'll tell you what it is in a minute if you don't already know. Well, we saw a number of plants that looked like this. Really narrow leaves, chlorotic, sometimes pink in color, sometimes coming up on recently planted rootstocks. Other times there would be plants that would have margins of the leaves like this. No evidence of a pathogen. You know, you can take your hand lens, look to see if there's any sporulation. Don't necessarily jive with any symptoms that, you know, you commonly read about. Here's a problem here that I saw on, I think, 2 or 3 farms. The thing that you notice here is the symptoms are in between the leaf veins. And usually if you have a fungus disease, the fungus grows across the veins. So anytime you see something where the symptoms are restricted just in between the veins, it's usually not a fungus disease. It might be more likely a bacterial disease or a physiological disease or maybe a virus disease. So I saw a bunch of this plant material like this and I collected it. So then we sent it off and had it tested for 16 different viruses that are potentially ones that you might expect to see on peonies. Well, when the results came back, the only virus that was detected on any of the plant material was tobacco rattle virus. And that is a virus that produces this very yellow, chlorotic, sometimes ring spots or a mottle on the leaves. It can be fairly small. And in some cases, you see this one where the plant has some necrotic areas as well. It's unclear to me whether there's more than one thing on this plant which is causing that. It's positive for the virus, but you could have more than one thing on some of these plants. Well, this is a virus that's nematode transmitted, and there are 2 strains of the virus as far as I'm aware of. One is primarily nematode transmitted. There's another strain that's nematode and sap transmitted, so mechanically. So you can see if you were harvesting flowers and potentially going down and you cut flowers off this plant and go to the next one, by sap transmission, that means that you potentially can transmit that by the contamination that might be on your cutting tool. Now, as a plant pathologist, normally we would say, if you have plants that have virus in them, take them out of your field. So I asked a bunch of growers in Western Washington, do they see this plant, this type of virus in Western Washington? Yes, they do. I've seen it and I asked them, well, what do they end up doing? Most of them, unless they're rootstocks producer, but if they're cut flower producers, are not roguing this out of their field. Their comments to me have been, they don't see it spreading that much. I don't know whether that would be true for growers up here. What I suggested to several growers last year, and I would encourage you to do that, to do what I'm going to say, if you have this disease in your field, take and map a small section of row and mark which plants on the map have symptoms. And then look at it over a couple-year period of time and see whether or not it increases. Because if it's transmitted on your cutting knife, you would expect it to be on the adjacent plants. You would expect it to spread down that row. If you don't see it spreading, maybe it's not that important. Yes, Pat?
I've mapped mine for about 8 years now and has never spread out of the plants that it started in.
So Pat's comment is that she has mapped hers for about 8 years and she's never seen it spread. One of the largest the largest cut flower peony grower in Western Washington, told me that when he first saw this in his field, he got very concerned about it, did some tests, decided not to rogue it out. He said basically it hasn't spread in his field. So I think part of it might be which strain of the virus you have. And Todd, I think you told me that there are both strains that have been detected here. It might depend a little bit on which strain you have, but that would be something, you know, that you could do to see whether or not it's something you should be concerned about. What are the potential things going on with some of these other symptoms that look like viruses? Well, a lot of times if I was a plant clinic person, a diagnostician, and a sample like this came in and I wasn't able to find a virus on it, I would say, oh, it looks like chemical damage. Could be. Well, when I was visiting a lot of growers up here, they weren't using chemicals. So then you say, oh, okay, well, they're not using chemicals, but what about residual chemicals, residue, if you will, or effects from applications of chemicals in the production of the rootstock, particularly like with Roundup. If some spot spraying had been done for Roundup and a portion of the plant had been sprayed, and you have this Roundup taken up and you see this effect. I've seen it in my own peonies in my backyard when I misapply Roundup and I happen to spray an emerging shoot. I'll see some symptoms very similar to this. So I just wonder whether some of this might be a carryover, and maybe Richard has some comments about this type of a symptom. The other thing that I noticed is, which is really sort of perplexing, is you'll get a plant like this where some of the shoot, when it comes out, the leaves will look perfectly normal, and then you'll have some over here that look just like this. And when I've talked to growers in Western Washington, they see the same type of a thing. But most of them are using chemicals, so it's a little hard to tell whether or not it is potentially a chemical residue type of a situation. And potentially, I think it could show up for maybe 2 years or so after an application of a chemical, if it was a systemic material like Roundup that was taken up. We'll probably do some tests to see whether that in fact is the case by treating some plants with some material like Roundup. So anyway, we don't know what causes this. I do think that there's some other things that were going on in some fields. A couple of the fields we're at, people were applying fertilizer mixes right around the bases of the plants. And particularly on small plants, you would see some of these shoots wilting and dying. We weren't able to isolate things from this, so I suspect that what's going on here is just pure fertilizer burn. Too much fertilizer being put on a concentrated amount of material right around that emerging shoot. You know, there's tremendous differences in regional and yearly environmental extremes, if you will, in some of these production areas. Last year was a tough year in some areas up here. Cold weather, soils maybe unprotected with snow, maybe poor drainage in some areas, and you have plants being killed, the root systems being killed. There was a number of plants and sites where I went to where plants had failed to emerge. You could dig down and there'd be part of the root system was rotten and had been killed, but there were still parts of the root that were still alive. And there were some shoots that were coming up even in mid to late July. So you do have this problem of having issues relating to sort of regional and yearly environmental extremes that you're likely to run into up here as you plant more and more peonies in some areas without really understanding how they respond to that. So let's look at some of the symptoms with the Botrytis. So it was interesting in that, at least from a pathologist, it's interesting. But we sort of have warped senses of humor and what is interesting and beautiful to us. But in some cases, there were these emerging shoots that were wilting at the top, had a blight, but the base of the shoot was still fine early on. You can see here, the base of this shoot is still green, although the top's been blighted back. Here, a little more wilted, and here it's been blighted all the way back down to the ground. So, a lot of Botrytis isolated from these. The more characteristic, if you will, foliar leaf spots and blight, even though it had been super dry and warm, you could still find Botrytis in plantings up here. And what was surprising to me a little bit is how much Botrytis was on some one-year-old first growing season crop in some of these fields. Suggests to me that some inoculum was being brought in potentially on planting stock. Or there may be another explanation. But in that, what would happen is you would have this blight go down the leaves into the stem and start growing back down this stem. So you have this shoot blight that you have the fungus growing into the stem. Don't know if you can see it, these little bands here on this shoot, that just shows the daily growth of the fungus. It's a growth rate. Wow. It's a daily growth rate. So it grew that much down the shoot. So think about it. If I have a shoot down here where I have the leaves infected and it gets into the stem, it's going to grow down towards that root system. And eventually it will grow down all the way to where it's coming out of that root. Now, I have not seen in the literature anything about Botrytis actually colonizing the root systems, but I'll tell you, what I saw up here would make me wonder about that. So this is a shoot blight coming down from the top back down to the base. Then we would see, or what I noticed is again wilting, but in some cases wilting of leaves that were green and you didn't see a blight on them. Here's a situation here where there's a single shoot wilting. Here the whole plant has wilted. It almost looks like if this was in our area, we'd think we had moles in there or something eating the base of the plant. And then in some cases, you have a situation where there were basically green foliage, but a discolored foliage on a single shoot or a cluster of shoots. And so you can see that here, here's an example here. And if you looked at these, in some cases, there'd be a dead shoot at the base. Well, if you pulled up some of these that maybe had the discolored foliage, but the shoot hadn't died all the way back, what you see here is some discoloration of the basal stem. So basal stem decay. It's not originating from the foliage and going down. It's originating from the soil or the roots and coming up. So if you had Phytophthora, which is a common problem on peonies in saturated soil conditions or poorly drained soil, this would be black. It causes a black decay of the stem. There's a reddish brown. One of the places that Harry took me to, the grower had sent a sample to Oregon State University, the plant clinic. They have a very good plant clinic there. Many of you, I think, have sent samples there, and the diagnosis came back as Rhizoctonia, which is a soil-borne pathogen and stuff. But I pulled up some plants, and I noticed this gray mold on the bottom of the stems, which is the common name for Botrytis is gray mold, because when it sporulates on colonized tissue, it produces this gray mass of fruiting structures called conidia on the surface. These are wind-dispersed spores that can then move to the foliage and cause infection when you have appropriate environmental conditions. So sure enough, all of the samples that we looked at that had this scenario all came back as a Botrytis. And then we also had these aborted buds. There's a number of things that can cause aborted buds. They could be directly infected by botrytis or other pathogens, or you can have environmental extremes that cause the buds to abort. But in many cases, we saw that these were colonized by botrytis. So, and then there are some situations where there was infections on the flower bud. So again, if you cut those flowers, put them in your storage, try to ship them someplace, you've got major issues. So as I said earlier, in relation to the flower infections, in Richard's comment about quality temperature controlled for storage, one of the reasons that that's extremely important is so that you do not have wild or big differences in temperature fluctuation where you get moisture condensation in storage. Because if you have big differences in temperature fluctuation, you have a cold product going into a warm product going back to a cold, you get condensation on the plant material, potentially that would allow something like botrytis, if there's spores there, to infect that tissue. Or you could have already infected tissue coming out of the field. So this just shows, a flower bud here that's been aborted. And I noticed that in some cases the leaves descending this bud were like this. This one, it's unclear, but in some cases there was infection. What I saw in the field was that there was infection starting on this leaf material down here. So as I've indicated, good temperature control is critical to reducing post-harvest losses, both from a quality of the product and from botrytis issues. So what about the diversity of botrytis species? Did we just find Botrytis cinerea and Botrytis peony? So this is all stuff that's done in a molecular lab where we extract DNA from all these isolates and run these tests, fingerprint them, sequence them, and fingerprint them, if you will, and look at where they fit here. We have, 200 or so isolates from various samples that we collected up here. Not all botrytis. There's Alternaria that was on some tissues. There's Cladosporium. But right now we've been focusing on sequencing and identifying all the botrytis isolates. So this is Katie Coates, who runs our molecular lab, and Monica is a graduate student who's working on this botrytis project. They're working on doing some isolation and sequencing. So what we found, as far as Botrytis peony, we found it on farms from all the way in the interior down to Homer, and a few farms in Washington. Remember, this is the botrytis that's specific to peonies, and generally is going to be quite aggressive on peonies. Probably doesn't need wounds and things like this to cause infections. Botrytis cinerea, much more common. Again, in Alaska, all the way from Fairbanks down to Homer, and in a number of Washington farms. I would have speculated that Botrytis cinerea would be more prevalent in plantings where flowering was occurring because of its tendency to cause infection on flowers. Flowers are extremely susceptible to Botrytis cinerea. Once you get infected, then you have sclerotia surviving from year to year, and I would expect an increase in potential problems. So here was the surprising thing this last year. You notice here, here's a botrytis. It's Botrytis sp. That just means it hasn't been named. And it's very close genetically to Botrytis convoluta, which is a species that gets on Rhizomatous iris and causes a rot of rhizomes of the iris. And it was picked up at a number of farms in the Fairbanks area. It's the only place we saw it. This probably didn't come in on planting stock. It appears to be a local indigenous population of an undescribed Botrytis species that has moved from native plants onto the peonies. And then there's Botrytis species that is very similar to Botrytis calthe, which I believe is a marsh marigold. And it was found in, we found it in 2012 at 2 farms in Alaska here. It was picked up in an additional farm this last year, and then this year we picked it up in a farm in Washington as well. So there's at least 4 species of Botrytis that we're seeing on these peonies, and as I said, the only reports in the literature are the Cinnaria and the Botrytis peony. And then, oh yes, one additional one here in Washington. So the symptoms associated with Botrytis cinnaria, you can see that basal stem decay, aborted buds, upper stem dieback, leaf blights, and foliar blight. A whole mash of the symptoms. In the case of Botrytis peony, mostly the basal stem decay, but also the foliar leaf blight, tip dieback, and the buds. The new Botrytis, the only place this has been previously found, one report in 2012 from grapes in Italy. Okay?
That's where we got it.
So that's where it came.
It's filling our wine on our
I don't know whether there's vineyards going in in the Fairbanks area, but with cuttings from Italy. But anyway, you know, on buds and leaves, none of the basal stem decay there. And if you look at the differences between Washington and Alaska, Cinnaria, about half of the isolates are Cinnaria in both in 2013. And then you see here in 2012, we see only these 2 species, and we have more species in Alaska. This shows the advantage of not necessarily just doing a single year's worth of surveys. You don't get the clear picture of what's going on. I'm not certain we have a clear picture now, but it's a very diverse picture. But when you look at these cultures, so we grow these cultures on media in the lab in dishes we call Petri plates. So it's a mixture of agar and various nutrients that these Botrytis species or other fungi will grow on. And you can just see right away, this is peony here, very small sclerotia. These black structures are sclerotia. They're hard, thick-walled structures that allow these Botrytis species to survive from one year to the next. Some have much larger sclerotia than others. There's obviously a much greater diversity of Botrytis species that have been reported in the literature. What the differences there are in the host-pathogen relationship of these is unclear. Does one require less moisture, more moisture for infection? Does one survive better or not? That's unclear at this point in time. I would like to shift a little bit here and talk about some general principles in managing Botrytis. Most of you may not care at all about the diversity of Botrytis species. You want to know, okay, how do I avoid this problem? Remember the quote from Alan Armitage? You need to know what you're dealing with. Accurate disease diagnosis is the very first thing. Sometimes pathogens can produce very similar symptoms, but they're very different pathogens. So if I'm trying to control Botrytis and I've got some information there, but I'm actually dealing with Cladosporium or Alternaria leaf blight or something like this, that management practice might be very different. So accurate disease diagnosis is important. Understanding the disease cycle of the pathogen, and I'll talk a little bit about that. Understanding factors that affect disease development. So as I mentioned, there are plant clinics where you can send samples. So there is a mechanism within the state And then there are other plant clinics. The one at OSU, as I mentioned. Puyallup, we have a plant clinic. There's a picture of Katie in our molecular lab. A lot of times when samples come to our plant clinic at Puyallup, we'll do molecular analysis for the clinic if they want to know species and things. The one thing that you'll find is that sometimes clinics are more specialized and more familiar with certain plants and hosts. I don't know if that's the case in Alaska. I suspect it is. I would encourage you, if your clinic or the people you normally deal with is not very familiar with your crop and your problems, don't just write them off. Encourage them to participate in field days and educate them and bring them along so that they're a resource available to you to help you locally. They're going to know more about the unique things that you're dealing with than anyone down in Washington and Oregon and stuff. But accurate disease diagnosis is extremely important. Understanding disease cycles and life cycles of pathogens is important from a standpoint that you begin to understand where I can affect the survival of this organism. This is a generalized disease cycle for Botrytis. Normally it survives over the winter time as the sclerotia. This is a very simplified one in that it only says that sclerotia can grow and produce spores, but sclerotia can also produce a perfect stage with another spore type, and it also says here that mycelium, it can survive over as mycelium in host tissues. Let's say maybe the roots of peonies. Under favorable conditions, it sporulates. These airborne spores are blown to the surface of leaves or flowers or berries. They germinate when there's free moisture. And the rate of germination and how long it takes for infection is dependent upon temperature and the host tissues. In the case of tulips, it takes about 4 hours. Once the spores are there, you have 4 hours of free moisture. With Botrytis tulipi, which is a very aggressive Botrytis on tulip, it's 4 hours. So it then begins to grow within the plant tissue. And it starts to produce another crop of spores, which are then blown to more leaves. And so you have this repeating cycle during the growing season. The length of time from when these spores land here to when the next crop under favorable conditions can be as short as 3 days. So if I apply a spray, how many of you think that fungicides kill fungal pathogens? No one? Okay. There are some materials, some biocides, where they will actually kill the pathogen that they come in direct contact with. But you can see here that a material like that might be effective in killing the inoculum or the structures on the surface of the plant, but they have no ability to affect the viability of the fungus already that's colonized inside the tissue. And fungicides are what most of the time are considered to be protected in nature. You have to put them on prior to infection to protect the plant from infection. You're not eradicating established infections. So that's the key here in thinking about managing diseases, is you want to minimize those early infections and keep them down as low as possible. So I'll use a couple of examples from some other bulb crops that I work on. So in this case, Botrytis blight on daffodils. Remember I told you about what fire, the common name, why it has the common name. So in this case, the Botrytis polyblastus, which causes this disease, survives over sclerotia. It only germinates to produce a perfect stage, which is a small mushroom on the sclerotia. These spores are only able to infect flowers. They can't infect anything else. On the flowers, the botrytis produces the conidia, which are blown to the leaves that infect the leaves. The leaves, once they're infected, produce more conidia, and you have this cycle. And I've been in fields that it's been less than 2 weeks from the onset of disease until the entire destruction of the field. So that's one type of a life cycle there. If we talk about tulips, Botrytis tulipi is a very important disease on tulips worldwide. It's the main reason you can't generally grow tulips year after year because of the survival of the sclerotia and the severity of disease. If it doesn't wipe out all the foliage, it'll cause infections on the flower petals, and you cannot market the cut flowers. This also survives, produces sclerotia on all this dead tissue. And you may not be able to see it, but there's small black dots. These are the sclerotia on the stem residues. Well, it turns out most of these Botrytis species can persist, the sclerotia can persist in the environment for about 1.5 years. They're not real long-lived. I work with some pathogens where the sclerotia survive for 10 years or longer. So in this case, they can survive for about 1.5 years. So these sclerotia that are produced here, if this debris is left on the soil surface, they'll sporulate the next spring when the crop comes up, and infections will take place. If the sclerotia are buried in the soil and the crop is coming up, and the emerging shoot here comes in contact with one of these sclerotia, it'll come out of the ground already infected. Or if the sclerotia are on the bulb, like this one right here, and this shoot comes up, this can infect that shoot coming out, and again, it'll be infected when it comes out of the ground. And it starts sporulating right away. These are called fireheads. So one of the keys in managing this disease is to remove fireheads early in the season. You just reduce the inoculum levels. Very similar thing happens, I think, with peonies. You get early infections. So removal of early infection tissue and stuff may be a way to minimize development of the problem on peonies.
Does flaming do anything?
So the question is, does flaming do anything? Flaming potentially would reduce the amount of tissue that survives, and and of course probably would kill the sclerotia that are directly flamed. If the sclerotia have already dropped out of the tissue down into the soil, the cracks and things, may not have much effect on those. And it doesn't take a lot of these sclerotia to be able to persist and bring the disease to the next year. Yes?
What stage in the life cycle is the botrytis most susceptible to damage itself? Where do you want to hit it?
So generally what you're doing is you're trying to do, and I'll make some comments about that in a minute. You're trying to deal with sanitation, getting rid of infected debris, and protecting the crop. You also need to be aware of disease development and the effect what I call disease pressure has on your ability to manage a disease such as botrytis. Pathologists like to talk about the disease triangle or the disease pyramid. You have pathogen, a susceptible host, and environment. If you don't have any, if one of these is missing, you don't have any disease. So if you can reduce pathogen levels, you reduce disease. If I have just a small overlap of these, then I have some disease. The more overlap I have, the greater the disease pressure and the more difficult it is to control the disease. And there's a time component. It takes time for the disease to build up. Once it gets started, it takes some time. Ideally, site selection is involved. Don't put a peony planting up against a timber stand where you're going to have prolonged periods of leaf wetness. You want good air circulation. Drainage, hills, things like this. Disease-free planting material. Host susceptibility. Understanding how susceptible is the host, and if you're monitoring, you monitor the most susceptible host cultivar you have. Don't be misled because you're looking at a cultivar that's only marginally or not very susceptible. Modification of the environmental conditions. That's really not feasible in many cases. There has been a lot of work with high tunnels, and high tunnels in many other cropping systems have been very effective in controlling and reducing botrytis infections because you reduce the period of leaf wetness, assuming that they're not overhead irrigated. Monitoring, sanitation is really important. Getting rid of that debris from one season to the next, removal of flower buds probably also, and then spraying to protect plants from infection. Let's see. I'll just make a couple of comments here about fungicides. I do a lot of fungicide testing because there's very few people who work on fungicides as it relates to ornamental bulb crops. IR4, which is a government federal agency to support registration on minor crops, has a program to look at reduced risk materials as well as biocontrol materials. In this here, this is a trial on tulips looking at botrytis. Here's this is thyme oil. It's an organic type of material. When there's an A above a column or columns that have the same letter are not statistically different from each other. So these are the most effective treatments here. There's no difference between the product, in this case the tulips that were sprayed versus the unsprayed, under fairly high disease pressure. In the case of botrytis on lilies, it only infects on the underside of the leaves. That's where the sprays have to go. If you're not spraying on the underside of the leaves, you're not really doing a very good job. So until we did that research, that was a really effective way for growers to be able to increase that. So if we look here, here's Proud 3 on lilies. And you can see that there is some reduction here in disease on lilies. But here's another organic-based material, and here's xeritol. Xeritol is an oxidizer, hydrogen peroxide. It's very effective in oxidizing and killing spores when it comes in contact, but it has no residual activity. So you'd have to be out there spraying this thing constantly to be able to get control if you were able to do that. So here, under light disease pressure, the Proud 3 had some promise. This is a problem with a lot of sort of these biological-based materials. They may work, but it's also going to depend very much on the disease pressure you have. So if you are going to use biological-based materials, and there are a number of them that are being marketed and stuff, just realize that you need to be careful about sort of the level of disease that you have and do a lot of other things to help manage that disease. A trial the following year on lilies, the Proud 3 worked fairly well the previous year, but under high disease pressure didn't do anything. So there's differences depending on the products you choose on how effective they might be. And you have to make those decisions based on your disease management program and the level of disease pressure you have. And I think with that, I will stop. I have a trial there on lilies. I do want to thank, though, particularly Pat, Harry, Sue, Clay, and Beth for their hospitality during my visit. They put me up, treated me very nicely. Gary Wells and Lydia Clayton and all the growers who were very kind and let me walk all over their fields and collect samples. And then for Beth and North Country Charters, who arranged a halibut fishing trip for me at the end before I got on a plane and flew home. So we're tight on time. I'll be around today, tomorrow. If there's questions, I'd be happy to answer them.